So welcome everyone to our next Dana-Farber targeted protein degradation webinar. And today is my greatest pleasure to host two speakers. Our first speaker is Stuti Mehta, and she is a scientist at the Boston Children's Hospital and an instructor at the Harvard Medical School. Stuti is broadly interested in understanding finer mechanistic details of gene expression controlled by transcription factors and epigenetic proteins, most recently by deploying targeted protein degradation approaches. At the Boston Children's Hospital, she worked with Stuart Orkin to use targeted protein degradation strategies to deplete the repressive zinc finger transcription factor BCL11A in service of inducing the curative fetal form of hemoglobin in B hemoglobin disorders. Here, she also leads a large-scale screening effort to identify glue-like chemicals that lead to the depletion of BCL11A. Stuti, we are very much looking forward to your seminar.
Thank you, Mikolai. I trust you can hear me.
Yes.
Okay. So, good morning, everyone, and thank you to the organizers for this opportunity to present our work to you. So I am going to talk about using targeted protein degradation techniques to understand a lot of biology about a transcription factor called BCL11A. The lessons we learned along the route doing this, and how we are using the learnings from this study to perform a high throughput screening for degraders of BCL11A. So I'll sort of zoom out and start with what a lot of you may know very well. This is the famous hemoglobin switch, and the crux of the hemoglobin switch is that the hemoglobin molecules that are in the red blood cells of newborns are not the same that are in the red blood cells of adults. So as you can see in this depiction, right before gestation and about 6 months into infancy, RBCs carry what is called fetal hemoglobin. And around 3 to 6 months of life, fetal hemoglobin is switched off. And it is replaced by the adult form of hemoglobin, shown here as red. Now, this switch in hemoglobin is actually affected at the genetic level. The fetal and the adult hemoglobin genes are expressed from the same cluster of genes, but from different genetic loci. And what happens in infancy is that the genes that express the fetal form of hemoglobin, the gamma globin, are shut off. And the genes for adult hemoglobin are activated. One of the most critical factors that gets this, repression of fetal hemoglobin going is BCL11A. And we know from a lot of work in Stu's lab and a lot of other colleagues that BCL11A acts as a repressive transcription factor by directly binding at the promoter region of gamma globin genes, and it recruits the co-repressor complex NERD to perform this function. And we know from a lot of work in the lab, as well as a few genetic clinical trials, that if you deplete the BCL11A protein, you can reactivate the fetal hemoglobin in humans and in mice. And we know that for patients of sickle cell disease and beta hemoglobinopathies who carry a defective form of beta hemoglobin, if you reactivate the silent gamma globin or the fetal globin, this can sort of cure symptoms of both these diseases. So as I said in my previous slide, fetal hemoglobin is curative of defective adult hemoglobin, and depleting the transcription factor BCL11A is a viable and tractable strategy to achieve this aim. Now, BCL11A is a canonical undruggable transcription factor. It doesn't have much of a structure to it that you can rationally design an inhibitor or a molecule to. It's basically 7 zinc fingers stitched together with IDRs. So we decided to look into targeted protein degradation techniques to target this transcription factor, because although gene therapy has had tremendous success for sickle cell disease and thalassemia. It does cost $2 million a pop, includes myeloablative chemotherapy, and is unlikely to be widely available anytime soon. So our questions in starting to deplete BCL11A by TPD strategies were twofold. We want to see if we can even degrade the transcription factor proteolytically. And the other question is, if we do achieve depletion of BCL11A, is this going to be a viable strategy? Is this going to lead to induction of the fetal hemoglobin? So I don't need to introduce this slide to this audience, but we do not have a binder to BCL11A, so we turn to the co-opted Degron strategy invented by Benam Nabet. This is so what we do is we fuse dTAG peptide to a protein of interest, which is BCL11A. And upon treatment with the dTAG-47 PROTAC molecule, this entire fusion protein is dragged to degradation to the cereblon E3 ligase machinery. Now, BCL11A has a dose-dependent effect on globin levels, on fetal globin levels. So if you it sort of acts as a rheostat. So the amount of BCL11A cell is actually quite consequential to its working. So we wanted to generate an fusion protein of BCL11A in erythroid cells at the endogenous locus. And after spending about a year trying to achieve this, we landed on an extremely efficient strategy to achieve HDR of large knock-ins, where we deliver the Cas9 and guide as an RNP, and the HDR template is delivered with an AAV virus. By the way, for cell lines, we use a crude preparation, so we've dispensed with the laborious purification for AAV. And at this point, we achieve anywhere between 10 to 40% knock-in efficiency. So I highly recommend this technique if anyone in the audience is thinking of performing a knock-in. So as you can see, there's a supershifted form of BCL11A. This is a fusion protein. And in less than 2 hours at sub-micromolar quantities of dTAG-47, we have an acute depletion of BCL11A. We also collaborated with Catherine Donovan at the FCI, and we see that this depletion strategy is fairly specific to BCL11A. By the way, we also tried 3 other co-opted degrons, which didn't work so well for BCL11A, at least. This was either because we couldn't get rid of the protein completely, or it took very high micromolar amounts of the drug, or very long treatments of about a day or so, which beats the purpose of trying to achieve acute degradation. So back to the dTAG system, we obtained several biallelically knocked-in clones of BCL11A FKBP fusion protein in HUDEP cells, which are erythroid cells. And we rigorously established that upon BCL11A depletion and dTAG-47 treatment, we do not see a growth or erythroid differentiation effects in these cells. And we used looking at the surface markers of erythroid differentiation for this purpose. Now, our other question is, can we upregulate fetal hemoglobin when you deplete BCL11A with this strategy? And to our sort of relief, we have generated sort of a drug-mediated toggle switch to control gamma expression, fetal hemoglobin expression. So when you add the drug, you deplete the repressor, the target HBG shoots up. When you wash out the drug, BCL11A reappears, you lose expression, so repression happens when you re-add the drug. So you can basically continuously keep adding the drug and washing out, and you will have toggle switch via BCL11A for fetal hemoglobin induction. Now, when we do cut and run experiments for BCL11A, we have about 5,000 peaks, and we get about 5,000 targets for BCL11A. And this strategy was our chance to figure out what are the direct targets of BCL11A, where it sits, and where it induces repression. So we combined acute depletion with nascent transcriptomics at 3 hours, and we see about 58 genes that are differentially expressed. This is at FDR less than 5%. We purposely did not apply a fold change cutoff here to allow for small differences in nascent transcription to accumulate into higher or meaningful differences in mRNA or protein accumulation. And then we charted nascent transcription in this in two different sets of experiments. The first set of experiments I'm showing you here is a sort of treatment of these cells with dTAG-47, so depletion for about 5 days, followed by washout of drugs. You can see BCL11A reappears within 24 hours of drug washout. And you can nicely see that when you treat the cells with the drug, the top targets sort of keep increasing in nascent transcription levels. And when you remove the drug, BCL11 reappears and you have a nice repression. And you can also immediately see the targets fall in about 4 clusters, whereas clusters 1, 2, and 3, you see a consistent sort of progressive induction of transcription and repression. Cluster 4 seems more like the noise in the system and possibly not direct targets of BCL11A. So we conclude that in erythroid cells, BCL11A targets an exclusive set of 31 genes, and that genes in cluster 1, which contain only the fetal and embryonic globins, are the most prominent responders to modulation of BCL11A activity. By the way, we also looked at chromatin accessibility via a tag-seq, and again, we see this sort of a hierarchy in responders, and we see that it's only genes in cluster 1 that ever lose chromatin compaction as measured by a tag-seq, whereas most of the genes in the other 2 clusters don't respond in terms of a tag-seq data. We also did a similar analysis in collaboration with Matthias Mann's lab in Germany to look at the total proteome. And you can see 2 days after treatment. Now we're looking a little further in differentiation and sort of further along in drug treatment because we want to capture differences in protein expression, which may take a little while to come about. So 2 days after treatment, and it's about 2 days after induction of globin, you can see the only genes that are upregulated here compared to DMSO controls are again the cluster 1 genes. They remain the most differentially expressed genes throughout differentiation. And you can also see that although the plot gets sort of dirtier, quote unquote, because there's secondary effects appearing, on the whole, when you compare the correlation of the quantitative proteome between dTAG treatment and control, there is a fairly good correlation. So BCL11A has a very specific set of targets. It doesn't affect differentiation when you deplete it. And the proteome looks very similar to control proteome. The other thing we can do with this system is, really look at how the gamma globin locus opens up when you remove BCL11A. And so far, all the information we have about the workings of BCL11A at the globin cluster have come from analysis of end states. So you know that in BCL11A knockout cells, you will have gamma expression. There's accumulation of mRNA. The chromatin is open, and there is loss of promoter DNA methylation. But in what in what steps are these changes taking place? So we chased acute depletion with analysis of nascent transcription, chromatin accessibility, histone modification, and promoter DNA methylation. And you can see that when you look at just the globin clusters, the induction of globin RNA is immediate and progressive 3 hours onwards. But you don't see that sort of a progressive opening of the cluster. And, of course, this is with the caveat that we are looking at the available resolution of a tag-seq versus pro-seq here. When you quantify, you can see this better. When you look at the maroon quantification of pro-seq signals, you get an immediate progressive induction of transcription. But at the current level of detection, we don't see such a progressive induction of chromatin accessibility. We also looked at changes in several histone modifications. The only 2 that we noticed a change was K4-trimethyl and K9 acetylation. And again, this takes about 24 to 48 hours. And this ties in very well with the cell cycle, sort of the division time of HUDEP cells. But the most surprising results we got from this analysis by far were the differences in, or no differences in, promoter DNA methylation at the HBG locus. So here we use bisulfide modification and Sanger sequencing to look at CpG methylation across 8 sites in the promoters of the gamma globin genes. And up until 9 days of treatment, when you have about 35% induction of gamma globin, which is above the curative levels of fetal hemoglobin induction you would need, you see that there is no loss of promoter DNA methylation at any of the 8 sites. So you can compare the red dots here, which is BCL11A depletion to DMSO treatment. Now, this is a huge surprise because there is an entire industry trying to strip DNA methylation from the gamma globin promoters to induce sort of curative levels of hemoglobin. So, from these results, we were thinking it seems that BCL11A silences the gamma globin promoters by a mechanism that is sort of parallel to induction of DNA methylation at the promoters. So we tested this hypothesis by co-treating our cells with dTAG-47 as well as an inhibitor of DNMT1 enzyme, which would strip DNA methylation. And I will not go into a lot of detail here, but we see that the contribution of BCL11A, the repressor sitting at the promoter, is far higher to repression of gamma globin compared to DNA methylation at the cluster. So from this analysis, we have sort of separated the epigenetic changes that take place at the gamma globin locus upon depletion of BCL11A. We know that transcription and accessibility and histone changes all sort of occur progressively and within the first 2 cell divisions. But the loss of DNA methylation that we see in BCL11A depleted cells is decidedly a secondary phenomenon. Now, what else can we learn from this system? We wanted to learn a bit more about, how BCL11A repression actually takes place at the locus. And we hear a lot about residence times of transcription factors on chromatin. And admittedly, a lot of this data comes from sort of positive transcription factors. But for BCL11A, time and again, it's been proposed that BCL11A is a hit and run transcription factor. So it's got fairly low residence times. And in some sense, it's not required, the presence of BCL11A is not required throughout sort of the cell cycle to repress, or continuous presence is not required for maintenance. So we hypothesized that if BCL11A is a hit and run transcription factor, the one time in a cell division cycle that it is absolutely required is on newly replicated DNA. And the way we can assess this hypothesis is that if you remove BCL11A in cells that are in S phase, you would see induction of gamma globin because you don't have any new BCL11A to establish repression on newly formed DNA. But if you look at cells in G1 phase, you will not see this induction. And you will have to wait for these cells to traverse the entire of S phase to finally be able to induce gamma globin. So we use the Fuji system to visually separate cells in distinct phases of the cell cycle. I won't go through this. This is published in 2008. It's called the Fuji system. And the experiment we designed in HUDEP cells was this. We determined the length of the S phase and the G1 phase in HUDEP cells. You can see these are both more than 7 hours. So we took 2 pools of asynchronously dividing BCL11A dTAG cells. One we treated with dTAG-47 at a high concentration, the other with a control. We continued the treat. We treated these cells for about 6 hours, so that's less than the time the cells take to traverse to the S or G1 phase. And then we collected the cells that were in late S or late G1 phase. So the rationale being that in dTAG treated cells, if the cells absolutely have to renew their DNA to lose BCL11A mediated repression, you will see sort of a high induction of gamma globin or high de-repression of gamma globin in late S phase. But because late G1 cells have not had the chance to go into S phase, they will not have induction of gamma globin. So first things first, in 2 replicates, this is DMSO, this is the bulk This is dTAG-treated cells, normalized DMSO controls, and you can see that throughout the 3 clusters, you have induction of all the genes. Within 6 hours, we also verified BCL11A degradation in a Western blot. And you can see non-targets don't respond to BCL11A depletion. When we looked at the fold change between late S and late G1 cells, disappointingly, we did not see higher induction of BCL11A targets in the S phase. And when you look at just the globin induction, you can see just as much globin is induced in late G1 phase cells compared to S cells. So we don't think BCL11A sort of only sits at the promoters to repress on new DNA. We absolutely think that the way BCL11A works is not by a hit and run phenomenon, and the presence of the repressor at the gamma globin locus, it's a large protein, is absolutely required to keep the active transcription factors out, and there is a continuous repression by BCL11A throughout the cell cycle in erythroid cells. So I'll summarize this part of the talk. We've established a quick and efficient knock-in protocol. You can look up our methods paper for this. We've also done a first generation PROTAC-mediated proteolytic depletion of BCL11A, and we've defined a core set of exclusively controlled genes in erythroid cells, and we show that the only genes that are sort of imminently and prominently responding to BCL11A loss are the embryonic and fetal globins. We've also established a hierarchy of the opening up of the globin locus, and we show that BCL11A represses the globin locus independently of DNA methylation. And in our opinion, repressor removal is absolutely required to induce curative levels of fetal hemoglobin. And we also think BCL11A is not a hit and run transcription factor. Apart from these mechanistic insights, our study has also provided us a proof of principle for proteolytic degradation of BCL11A, and we've designed cellular dual-luciferase based screen, which I will introduce you to today, and this is very much ongoing. So this is a dual-luciferase screen. We are performing this in HUDEP cells, which are erythroid cells of human origin, and all this work has been done on. Here we are using a fusion of BCL11A and nanoluciferase. So nanoluciferase is the proxy for BCL11A abundance in the cell. And the reference luciferase is Firefly. So this is a control for toxicity. This again is being done at the endogenous locus because, like I said, BCL11A has a dose-dependent effect on globin induction. So if we achieve, a certain amount, not complete depletion of BCL11A, we still want to be able to catch that in a physiologically relevant frame. This is sort of an example of the kind of z-scores we are achieving with this screen. If you look at the right y-axis, this is plotting of the z-scores. The screen is extremely good at being able to tell a true positive from a negative. So in the screen, we have plotted the ratio of nanoluciferase, which is the proxy for BCL11A, and the reference Firefly. The red is a positive control, dark blue are DMSO controls. And each light blue dot is a compound. This is about 50,000 compounds. And the rubric we are using to tell hits from non-hits is if you have a reduction in nanoluciferase and the ratio of N by F goes down below DMSO, we are calling these hits. So this is also a pretty clean screen. We are getting about a 0.3% hit rate. We are carrying out, which I just found out is apparently called an ultra high throughput screen with, we've processed about 300,000 compounds now from various sources. Most of this is on a collaboration basis. And a couple of these avenues we're particularly excited about are the electrophilic binders and the natural product extracts, which we're working with John Clardy at the medical school and the University of Michigan Natural Product Core. While we conducted all the screening, we also realized that you're always screening for a fusion protein. And so what you're looking for is both molecules that target BCL11A as well as Nluc. So we designed a counter screen here. We simply introduced a P2A cassette to separate the two proteins in this system. So this is our counter screen. So anything that passes the primary, we take it to the counter screen. In this system, if you have a compound that reduces the nanoluciferase levels, you know it's targeting nanoluciferase and not BCL11, and you can eliminate those compounds easily. The other thing this counter screen does is it eliminates transcriptional hits, because both the proteins are produced as in a sort of bicistronic mRNA. So if you have a reduction in transcription, we don't want transcriptional hits. By the way, they're much more pleiotropic. So you can eliminate transcriptional hits this way as well. And this is going to be my last slide. Like I said, this is very much ongoing. And then we plot results from our primary screen to the counter screen. And you can sort of tell that the compounds we're interested in would be the dark blue ones, which reduce nanoluciferase in the primary, but don't have much of an effect in the counter screen. And we are following this up with sort of low throughput methods, Western blots, and always controlling for cell viability. And we will be talking more about this if and when we find any valid targets. And we're constantly sort of looking at the hits, but we're also constantly thinking about the biology of BCL11A. A lot remains to be understood about the protein. And one of the biggest questions really is, what's the endogenous degradation pathway for BCL11A? If we understand that, it will help us design sort of a rational targeter to BCL11A. And with that, several people I have to acknowledge. I work in the lab of Stu Orkin, who is an amazing mentor. My technician, Alta Buenbat. A lot of the bioinformatics was done by Yan and Chen. I'm working with Linus and Duigu on the screen. Frank is our lab manager, and several collaborators. And I may have taken a lot of time, but I'm ready for questions.